Another tool that's used to further the field of microbiology is the scientific method. And I know this isn't a typical like physical tool like a microscope, but it is a tool that we can use in our thinking and our way of approaching the problem. And so the scientific method starts with you formulating a question. Maybe you look at something and observe it and go, wait a second, I wonder why that works that way. You come up with a question. It needs to be testable. Things like you need to know what organism you're looking at and what you're going to be measuring. Then you do some background research on it and try to fine tune it and design an experiment. You'll construct a hypothesis of how you think it's going to go, what you're expecting to find. Then you'll test the hypothesis and you'll have a control group and an experimental group, and you'll compare them, analyze the data, and determine, was my hypothesis correct or do I need to do some more work? And then if your hypothesis was correct and you validated it, then you're going to go on and communicate the results and come up with a new question. Or if it wasn't, it's just as valuable. You'll come back here and rethink your question and maybe design a new experiment to try again. This is a great tool that assists us in studying the natural world. It's not the only way of knowing things. A lot of scientists would tell you it's the only way to know things is the scientific method. And while it has value, it's not the only way. We can know things through historical evidence. We can know things by our own observations. We can know things, since we're at ORU here, through the work of the Holy Spirit in our lives. But this is a great tool to begin to test and tease out exactly why things work the way they do. A couple other tools that have advanced microbiology are restriction enzymes. So these are small kind of scissor-like enzymes, basically. They can come in here and they can cut very specifically DNA. See where the little scissor sites are, and they'll make sticky ends. You'll see the overhang here. Cut it open, and we can use this to basically paste in other genes to use in our research. Okay, so it's a cut and paste sort of option where you want to add in another gene to make insulin, like my earlier example. You could cut it open here with a restriction enzyme, glue the insulin gene in, and make more of it. Another tool, PCR, is a copying tool, basically. So polymerase chain reaction, or PCR, is used to amplify or make copies of DNA. So you can start out with your DNA of interest here and add a new strand to it by heating and cooling it. You can get multiple copies of it and they'll be identical. So you'll end up with your copy of the insulin gene over and over and over again until you get thousands of copies, enough that you can do more experiments on it or study it more fully. Another advance is in our understanding of RNA. Okay, so we're getting a little ahead of ourselves here, okay? But there's this idea of the central dogma of biology. And this states that you have DNA, our double-stranded genetic material, and it gets used to make single-stranded RNA that then gets used to make proteins. And typically that's what we think of with RNA or mRNA, messenger RNA. But in recent years, we've discovered that there are multiple other types of RNA within the cell. We have microRNAs, short interfering RNAs, short hairpin RNAs, long non-coding RNAs, aptamers, all different kinds, all that are kind of regulating the activity of the cell. So these ones can silence genes, they can edit genes, turn them off, act as little machines. All of these sorts of things can help kind of turn up and down gene expression or modify the activity of the cell. Another advance is in genetic sequencing. So in the early thousands, we completed the Human Genome Project, which its goal was to sequence all the A's, T's, C's, and G's, all the letters of the DNA code for humanity. And they completed that. Now we have all of our chromosomes sequenced, and you can go on the government database and compare them to your own. And the next step in this project is to not only have our own genome sequenced, but to sequence the human microbiome. This is the Human Microbiome Project. And the idea behind this is, can we identify and sequence the microbes that are a normal part of our microenvironment? So what organisms, what bacteria are normally found in our gut, or what are normally found on our skin or in our eyes? And can we characterize them and figure out what proteins are they making? What are they doing? How many of them are there? What makes a normal microbiome and which ones shouldn't be there? So maybe we can identify the problems before they show up. Another big discovery in the field of microbiology was the discovery of spores and how to sterilize them. Endospores are heat-resistant dormant bacteria. Okay, so you can kind of think of them as a seed to a plant. So a seed is still living and the plant is living, but the seed is the dormant version. Same thing with endospores. They are the dormant version of the bacteria. They're not multiplying or growing. They're just kind of hunkered down until conditions improve. And these were discovered by Ferdinand Kahn, who found that heat would kill most bacteria, most vegetative, normal bacteria that are metabolically active. He could kill them with heat. So in the microbiology lab, we often use metal loops to move bacteria different places. And he found you could heat that loop and it would kill most of the bacteria. But the caveat is it won't eliminate endospores. So it will kill the normal vegetative cells, but these dormant bacteria are much more resistant. To kill them, we need to sterilize. And to sterilize is completely free, whatever surface it is, of all life forms. This includes spores, viruses, bacteria, archaea, all gone. And one of the ways we do this is with an autoclave shown here. So it has little metal trays. You can close the door on it. It gets super, super hot with high pressure. These are often found, like the small versions, in like dentist's office, where they will sterilize all of their metal tools. You might see them. I worked in an eye doctor. We would put any of the tools we worked with patients with in a sterilizing autoclave like this. But we can sterilize any of our tools we're going to use with patients or for research to get rid of endospores and all living particles. Another development for furthering research and work with patients is the development of aseptic technique. So in the mid-1800s, Oliver Wendell Holmes was doing some research at his clinic and found there were two hospitals in the city that serviced women's health. And one was having more infections than the other one. And he was also finding that if women gave birth at home, they had less infections after giving birth than those that went to the hospital, which is pretty discouraging if you're trying to get people to come to your hospital, have your baby here, and they're going, wait a second, no, I'm going to be in worse shape than if I just stayed home. Okay, so he made this evaluation and didn't quite know what to make of it, but it was an observation. Okay, a few years later, another doctor, Samuel Weiss, showed that women were becoming infected at his maternity ward after being examined by his student doctors. Okay, so he found that he had multiple student doctors, and they would go and work in the autopsy room, looking basically the morgue and doing autopsies to see why did they die. And then they would go without washing their hands to the room next door and help deliver a baby. And then they were scratching their heads as to why this was a problem. I know, very shocking. Okay, but this was a huge discovery at the time to see, wait a second, I might be carrying something I can't see from one room to another. And the person that put this together was Joseph Lister, who showed that if you used aseptic technique, disinfected your hands, used chemicals to clean the area, we could decrease infections. Okay, so at his hospital, he introduced this idea of, okay, if you're going from the morgue to a patient, you have to wash your hands. And he would also, as you see in this picture, clean the air around where they were, the surgical site, to keep anything that he couldn't see from hurting the patient. Okay, so aseptic technique is the method of handling microbial cultures, or it could be patient specimens, anything that you think might have microbes on it, in a way that prevents infection. And so this is important that we take care of the sample, ourselves, and others that could be exposed. So this would mean in the microbiology lab that we wear gloves and we sterilize everything. It could also mean if you're collecting blood samples for a patient, that you collect them in a sterile manner and you store them in a sterile manner. You don't, I don't know, dump all of the blood samples together. You keep them all separate with the cap neatly on them. And same thing with dealing with the patients themselves. This has been a big deal with COVID of, okay, we're going to put all of our PPE on, and then we're going to take it off and put new PPE on before we see our next patient, all to keep infections at bay. Okay, so our last one of the day here is the germ theory of disease. This is a theory from the 1900s that has been followed up even into recent years. The idea that we can figure out the exact microorganism that is causing the exact disease. This was started by Louis Pasteur, our guy we was talking about earlier with the crook-necked experiment and the vaccines. He showed that there were certain microbes involved in winemaking, certain ones in pasteurization, and was starting to tease it out. But the person that really followed this through was Robert Koch, and he established some postulates or series of logical steps, a flow chart basically of ideas that you can use to determine what organism is the problem, which one is pathogenic, which one is causing the disease. And he did this originally for anthrax and showed that anthrax is caused by Bacillus anthracis. Okay, so here's how the flow would work. You'd start out with you think Bacillus anthracis is the problem. Okay, so you'd give it to your little lab mouse here, and the lab mouse wouldn't go so well for a while, and eventually you would have a sad dead mouse to deal with. Okay, so you have your mouse here, and from it you would collect microorganisms to grow on a plate here. Okay, so microorganisms are grown in pure culture. Pure culture means you're trying to isolate and get that one pathogen by itself. Okay, so you'd grow it up. You'd also look at it on the microscope, see what shape it is, maybe do some staining on it to visually evaluate it. And then you would inject a healthy animal with what you've isolated and see if the disease progresses in the same way. So if the first time over here, it's a lung disease and now it's a skin disease, we go, wait a second, I don't think we have the right pathogen. But maybe you're going, okay, well, last time it had a weird rash and now we have that same exact weird rash back again. Okay, so seeing that it manifests the same way. And again, we collect the microorganism, look at it on the microscope, And if everything checks out, then we can determine, yes, this is the microbe that causes this disease. Okay, so that takes us halfway through chapter 1. I will see you again next time.